choose your.
Yeah, choose.
All right, has everyone got the invitation for the open review reading assignment? Okay, cool. All right, let's get started. So last time we talked about how AlphaFold has been evolving to predict protein from single sequence with some MS information to the structure of the protein, which we call protein folding. And of course, there are additional like molecular system other than proteins, such as DNA and DNA, RNA, and small molecules, which AlphaFold 3 and a bunch of follow-ups of AlphaFold was trying to tackle. So you can see that at the end of the day, what we care, the reason we want to predict protein structure is still like the final goal of this pipeline is not just getting the structure itself, but rather try to understand the functionality of the protein. And we've seen this kind of central dogma multiple times, but what actually drives the molecular function of the protein is the interactions between the molecules, between both either proteins or protein and other ligand like DNA and small molecules. So after we were able to predict the structure in some degree, a very natural mindset would be to look into like how well we could further extend the modeling of single protein. Sometimes we call it like monomer proteins to a multimer system or complex systems, where we at first time will begin to like know look deep into the interaction mechanism, which determines the protein function in different ways. So a lot of the molecular functions, right, and biological functions, like for example, like proliferism or metabolic process or just signaling pathways rely on the interactions between protein and proteins. And such, so basically from the last time, we mostly focusing on, like if you give me a single chain here I means like one connected polypeptide, which usually fold into kind of a single domain or a single unit of the proteins. And so from this lecture on, we're going to talk about protein interactions, which involves multiple chains of proteins, where we can consider multiple subunits. Each of them is folded from a single polypeptide or a single chain from the DNA, and they need to kind of interact and assemble or intertwine in some way such that their interaction pattern will decide the unique functionality of the protein. All right, so I think we would talk about interactions, but most of the time it could be all kind of boiled down into one fundamental phenomenon, which we kind of call it like association dissociation, which basically considering in the chemical reaction where on the left-hand side you have two monomer proteins or two like different parts of the of this dynamic systems, and on the right-hand side is basically a potential like product that the two separate free systems will intertwine and become like a new product. So I think the interactions we're talking about, a lot of time you can hear different words, like sometimes like binding energy or binding affinity or binding reaction rates. They're kind of sort of all connected. So when we talk about interaction, we almost always talk about or hinting at the binding affinity, which is kind of determined by a dynamic system where we care here about basically how much of the left-hand side will maintain its form in the left-hand side versus the right-hand side, and what's the rate between the two. What's kind of the rate of you see like the system what kind of outcome you observed when you mix A and B together into a system, and the equilibrium here means that because every reaction happens in two directions. So if it happens long enough, it mixes long enough, there's kind of the sort of equilibrium where you could assume that if you observe the kind of density or like titration of the single molecules, it will maintain at a certain level, even though like they're constantly changing from left-hand side to the right-hand side and from right-hand side to the left-hand side. But if it happens at a certain rate with like fixed speed or constant, you will kind of observe a kind of dynamically equilibrium system where we will be able to still compute certain constant for measuring like how stable this reaction is happening and how likely that you will observe the stable form of like bind molecules on the right-hand side. So very important quantity related to this process. Of course, this kind of sometimes people call it like kmol or reaction rate, is one way to characterize this interactions. And the other ways of categorizing it is perhaps also looking into the entropy and the energy of the systems. And one type of energy that we care about is the Gibbs free energies, or sometimes we can see delta G or delta G, which like in high level talks about like how much energy is changed when the two molecules stay in the unbounded state versus in the bounded state. So I think when they're in the unbounded free form, we call it apple, and when they bind, we call it hollow state, hollow state. So you can consider delta G being kind of the energy gap between the two states, and they are all like very related to each other. So kind of there are the whole like binding interaction prediction is basically trying to mimic or try to use machine learning to model these entities, but with different level of precisions and with different amount of degree of freedom. So the binding affinity is a very general concept, but in the real function, in the real world, the function of the protein inside the cell can have very different types, but they're mainly differ in terms of like the strength of their binding and the lifetime of their binding. So there are a lot of kind of micro scale, smaller than micro scale lifetime reactions that almost happening in a random basis, which we almost never able to capture, or like if we're capturing it, they're mostly like commonly existing patterns that only statistically making sense and very non-specific. So some of such patterns could be like packing, where like really weak interactions that try to like organize the protein in some really weak fashion, and they can be very, very transient or like short term. And there's some like from a little bit short-lived to transient interactions, and these can be think of as like the protein goes through some intermediate state to be able to carry out certain function, but after that they can go back to their original state. And one typical example of that is enzyme, right? So enzyme and substrate, they kind of intertwine in the middle, and when the substrate for the substrate to react, and the substrate will become kind of jump into the intermediate state, but at the end you get the reactant, the product out, and their enzyme will stay intact, and they can be reused for the next reaction. Similarly, for example, like for the transduction of signals, right? In the signal pathways, you have receptors and signal carriers. Like they sometimes like you just want to trigger a signal like for a very short term, like a few seconds, and that's enough to like open up the pathway and passing this signal on. So that kind of interaction are also like transient. And there are certain interactions that are much more stable and also last longer. Some of them can last almost like days. And when we measure their kind of KD, which is the association constant, it can be at the picomolar scale, which means that it's really tight and solid binding that's happening, which is very ideal for kind of drug-like properties like antibody-antigen binding or like inhibitor binding. So if you design binder, sometimes we mean that we design something at like from the stable to permanent range. So from a nanomolar to picomolar range, so those things can really stay long enough and form like oligomeric proteins, which is kind of a complex that it assembles multiple monomer proteins. And even though like these transient interactions happens with like weaker at a weaker like KD, but they at this range they could already be pretty specific. But again, like for these kind of reactions, sometimes we want it to be very specific, so we don't catalyze very different types of reaction. But sometimes we also want it to be pan substrate, for example, one thing that can work for multiple targets. So specificity is something that really depends on what target we're designing for. All right, so when we talk about protein assembly and protein complex, especially for proteins that are stably binding into oligomers, we have some nomenclatures describing these complexes. For example, like basically depending on how many subunits are binding to each other, you could have this interfaces that consists of different numbers of monomers. So if you have two monomers interacting, you call it like dimer. And if you have like 4 of the monomers to form one assembly, then you call it tetramers. And similarly, there are like different numbers of monomers interacting to form these larger complexes. And depending on whether you have a single type of monomer proteins to form this oligomer, sometimes you can see like people say homo oligomer and hetero oligomer, where homo oligomer means basically you're repeating the exact same subunit multiple times to form this like oligomers, versus hetero oligomers are involving multiple pieces of different subunits. But again, like even a single unit can repeat multiple times. So in this case, you have like one of them repeating twice and the other also repeating twice to form one complex. So this is a really dynamic and very diverse process where you could see you can make use of different materials to combine them together to make like bigger proteins with different symmetries and different architectures, which is a very fascinating and difficult area. And particularly, question?
Only a naive question. What's the difference between like a multimer and an oligomer?
I think multimer is basically the same thing. I think multimer is like a kind of easy to understand, kind of common plain language name for oligomer. Yeah. So like alpha-fold multimer, it means like or you can think about multimer being not just between proteins, but could be between like multiple molecules with different molecular systems. But for oligomer, usually we mean protein, like protein-protein interactions and things that form by protein itself. And you will see the word heterodimer a lot because heterodimers are the most commonly used system to predict for a binding. Heterodimers means you have two different proteins and you have two of them, and you just predict like how they interact with each other. So a lot of the data set that is trained for this protein interest involves heterodimers. So we also call, I think I mentioned a little bit before, but kind of each single subunit that come from the same polypeptide, we call it a chain. And usually this, you can consider chain being kind of an independently transcribed subunit, or yeah, it's kind of talking about sequence level, but basically it's a kind of a single amino acid strain that can be folded independently, and they can form the different domains and functional unit of a protein. So the concept of chain exists when you look at the PDB files for a protein. There will be a column called chain that kind of try to give you this segmentation of like small domains and contiguous peptides. All right, so when we talk about interactions, actually not just protein-protein interactions, but protein-ligand interaction, where ligand can be any other thing like DNAs or small molecules. We're actually talking about a lot of different tasks, and these tasks have, as I mentioned earlier, very different level of precision needed, and also like different level of data that exist in the world, such that making each of these models for different predictions will take slightly different design choices, and based on like the kind of the easiness of the task and the data available there. So I've tried to categorize the tasks into kind of these pipelines of 4 steps. Right, the first is kind of very simple. You can consider it a simple one, but this is the one that like is broadly, the data is more broadly available, is whether there's interaction or not. Basically, for any given pairs of proteins with very minimum information, I would just try to say, like, do a binary classification and say if there's any interactions. Or you can do some regression, but usually that's very hard at this stage. So basically, just give me 2 proteins with minimum information, how much we can predict for like their interaction patterns. So this happens kind of, we can consider like a lot of different bioinformatics tools being used at this stage, not necessarily machine learning, but we can make use of these huge networks and this kind of low precision data that we get from measuring the interaction of patterns with kind of a binary precision, and we actually have a lot of these kind of binary level information about interactions, which forms this commonly known like protein-protein interaction networks. Right, so the task here is really kind of how we make use of this basic information of proteins, such as their family name or their sequence, and as well as combining with certain like relational structures, such as protein-protein interaction networks, to make these predictions of like general like binary protein-protein interactions. And the next level of the task is basically one step further. Say if I know that 2 proteins are binding, where are they binding? This is still like not completely like going into the very precise 3D composition of the structures, but in a high level, I want to just predict like in the whole protein which part of it engaged in this interaction, and therefore it would be important to look into these positions. And sometimes that will also maybe predicting this step is already enough, because when we try to design the interface, we're most likely designing these residues of the protein. So even without going further into downstream tasks, we may just stop here and also make use of these kind of prediction tools to guide us towards how we design or how we can like choose pairs of proteins to bind. So some of the data, some of the input feature used here can be both like sequence level information or evolutionary information, and also sometimes like structure information. And once we have this, we tackle this stage, that will give us a lot of good information for the downstream tasks as well. Particularly because 2 proteins, they have too many small surface in their inner surface. So basically, when we try to do more fine-grained interaction study, we need to resolve the first degree of freedom is where the contact, right? So because you like if you have two things, you can like have them contact in very different ways, but there's maybe only one or two, a few of them is actually like very low energy and very stable. So knowing where the interface is can save us a lot of compute and degree of freedom for downstream task of precisely predicting the interaction of the protein and their structures. So as an immediate next step, I think the task 3 and 4, they sometimes can be kind of we can consider them as at the same level of difficulties, but I would usually like mention task 3 first because I think that's kind of like a very immediate step following task 2, which is instead of just knowing where the interaction happens, I also want to precisely know where they are when they interact and form a complex. So this is like the 3D conformation prediction of a binded protein complex or protein ligand complex in their bounded state. And this is what usually people call docking. So basically giving two separate molecules in their free form, we try to tackle the problem of how these two molecules binds together, like their relative translation rotation with respect to each other, such that they can form the final like stable complex. And kind of knowing where the interface are gives us some hint about where they combine, but that's not enough because like the distances between the surface is like it's not fixed yet. So at this step, we need like much higher precision to basically predict the precise location of the interface atom and their orientation. So some of the models that solve these problems with machine learning, including, for example, the AlphaFold multimers and AlphaFold 3 for protein systems. And there are also like dedicated systems for solving the small molecule and protein binding problems such as DiffDock and Equidoff. So finally, the task 4, I consider this being the most difficult task, which is exactly directly predicting binding energy and precisely like accurately predicting binding affinity, which I feel like even right now it's still a very hard thing. So you can interpret this as like understanding the whole like biophysical process of docking and binding, such that if we can predict the energy well, we basically can solve all of the first 3 tasks, right? And if we're able to kind of predict the binding well locally, that will also help us to design a better protein, right? Making small changes locally, such that each time you make a change, you locally improve the binding affinity. And this has also happened a lot in the current like antibody or protein optimization, protein engineering pipeline, where you just try to propose small mutations and small changes to a starting point. And we call this kind of affinity maturation. And the question here is whether we could build a model that predicts this energy or binding affinity, the local landscape of that very well, and given some initial like structure information and pre-existing data. So in my point of view, I think these 4 tasks, I organize them in the order from like the reliance on the structure, where the further down to the right, we rely significantly more on the structure information. And also the precision of the modeling will become higher and higher, and the existing amount of data is also becoming lower and lower, right? So for example, for the left-hand side, if you just care about interaction, whether there's interaction at all, we can use a lot of the high-throughput approach to give us already pretty accurate profiling of multiple proteins against multiple proteins. Versus in the middle stage, you will basically need some level of structure information. You kind of need to solve some complex in crystal form. And the task 4 is like even harder, where you have to measure this KCAT association constant, which usually also can only be done with very low throughput assays. Any questions so far? All right, so we'll start from the left-hand side first. So task 1, just in general protein interactions. So as I mentioned, if we just care about like knowing about interactions within all the like kind of naturally existing cells, existing proteins in like cells, right? We kind of already know the whole genome information of many species, including human, and we were able to kind of already plot like a network of proteins, right, based on their kind of functionality. And understanding these interactions just at a binary level would largely help us to further help us draft out the pathways and how give us kind of systematic mapping of how the proteins interact inside the cell. And so the most precise information we can get is the crystallized structure of the complex. So if something that binds already in nature, they most likely exist in a complex form. So we can crystallize them together and solve high resolution structures. But those kind of data are, again, kind of limited comparing to the other kind of sequence data that are available. So alternatively, there are a lot of high throughput screening approach that can be used to screen for if you have a list of multiple candidate proteins and just want to profile the interactions. There are a way of coming up with this multiplex assays where you could at a high level measure if something bind or not. And there are multiple such type of high throughput assays. But I think in general, I think this Yeast-2 hybrid and mass spectrum are very commonly used. But a lot of these is just try to, again, make use of the sequencing information to predict whether something interact or not. So take this Yeast-2 hybrid assay as an example, right? So the way it acts is basically you have this reporter gene where if you have a transcription factor that's functioning in the non-coding region, the gene will be activated and be transcribed. And you can measure this by measuring the mRNA number of recounts for the mRNAs. So how they set up the system is you basically have two proteins that you want to test for the interactions, and you kind of hybrid them with one domain of the transcription factors. And if you can put a lot of these pairs into systems, and if the pairs bind, then you will get all of the functional domain of these transcription factors, which will effectively activate the transcription process of the downstream reporter gene. So with the sequencing technique we talked about in the first module, we'll be able to measure whether these pairs of protein interacted, basically is there interaction or not, by just measuring this reporter gene expressibility. So you can see that this is not a measurement of exactly binding affinity. It's kind of give you just high-level information of whether these two things bind and triggers transcription or not. So using a lot of similar kind of techniques exist, but using such kind of techniques, we the scientists were able to actually profile a lot of the interactions between known proteins for multiple species, from most well-studied yeast, fruit flies to even human. There's some data, some amount of data already exists for several species where kind of you can plot these complex networks between all their transcriptome or proteomes and define what we call interactome data. So interactome means the connectivity of proteins inside a given species proteome. Proteome means all of the proteins that one species can express. And interactome means their interaction patterns, which is kind of a very good source of data that help us understanding the biochemistry of how cell functions. So by using the high throughput experiment we talked about previously, we kind of already have some amount of information about these protein-protein interaction networks. And even though we don't know how precisely how the interaction happens and how strong the binding affinity and their precise structure of binded complex. So then here, so this you can consider this as kind of a preliminary data for training a model to learn from either just from the pair of interactions or from the entire network information, and we can tackle we can build a model to see how well we could model the system and predict for any given two proteins, are we able to predict their interaction existed at all, yes or no problem. So I kind of like this diagram where were kind of one. I think a professor tried to kind of categorize different models in model-input interactions, but on this like x-axis, it's exactly like whether we just care about the yes or no questions, or we want to get a high level, like more detailed information. So, so in the very end of this axis, there's kind of the very preliminary sequence-based deep learning models, which are enough to make this kind of predictions where, like, if we give you 2 proteins and their sequence, we might be able to already predict something from the sequence itself. And such data exist broadly in the nature, and not all of them we have a ground truth structure for. So basically, sequence-based protein-protein interaction prediction task is can be simplified as like give me 2 sequence of 2 proteins. We build a model to predict whether they bind or not. So it's very straightforward. And for example, models like Descript from MIT group, Bonneberger's group, they just make use of like pre-trained language models where we can basically convert the amino acid into pre-trained embedding space, and we can make pairwise, computations between pairwise embeddings. And basically, this is like the pair matrix that you see in like the AlphaFold, and we can use that to kind of. This is not exactly what it's predicting, but it is kind of like interpretation of if you train a binding predictor, what might be the intermediate layers learning, right? So the final outcome is still some integrated prediction module. You can consider all kind of pooling or multi-layer pooling architecture to get you to the final like logics of predicting whether it binds or not. And you can just train this with cross entropy loss as like classification models, and at the end, like this method to be working reasonably well, and they also make the interpretability tools to look inside these high-level prediction, high-level like activation maps, and observe some level of correspondence between that and the actual contact map, like where the protein binds. So so this is just using the sequence data, but as we saw that like. Beyond this sequence data, there is also already like existing network data that we can kind of profile from this high throughput assays, right? So assuming that you have like some connectivity given part of the network, and a simple idea would be like if I know the network structure, maybe I'm able to like interpolate or extrapolate beyond that, so such that I can just like for any 2 proteins, I can just look into the network and see their interaction patterns, and maybe I can guess whether there is a direct, interaction between the 2. So a broad range, like, so yeah, actually a lot of the previous bioinformatics tools for predicting these interactions are basing on these kind of network analysis, where people come up with different features, featurizations, and descriptions of the neighborhood structure and topology, like using some complex algorithms. And with these features, they can try to make predictions of whether there might be some interactions happening. So a kind of a very, very natural like downstream thought is just to combine both the strengths of language models, like the sequence level information, and this topological information at the protein-protein level, and like just make the combination of the 2, such as we basically can have this augmentation loss where instead of just predicting from sequence, we can also have this augmentation loss that is used to like boost our prediction power. And this kind of method works much better than the like just relying on single sequence predictions. And as we're getting better and better at predicting the structures, right? And a natural information that we want to leverage is like also the structure information. So there's also kind of this more recent work called high PPI, for example. Of course, there's probably 10 other works that works on similar topics, but basically you try to make use of all existing information, right? And including like the protein sequence, the protein structures, and protein subdomains, and their community level, which is the network level information. And what they did is just kind of making these multi-level graph neural networks, where from the bottom you can model these very precise geometric like neural networks that learns about the geometric features of your protein structures. And you can also make use of pre-trained language model for embeddings. And at the end, you get these kind of embeddings that you can use as the node features. And you can put it back into a protein-protein level interaction network and train another layer of graph neural networks on top of that. So at the end, you kind of get a very integrated and multi-level relational data that you use to address these interaction problems. Yeah, a lot of similar work exists. So if you're interested, you can definitely search around with these keywords, and there are a lot of interesting work on that regime. So, any questions so far?
Just to clarify, the protein-protein interaction, that's just for direct interaction with proteins? It wouldn't include like indirect correlations or intermediate signaling molecules causing interaction?
I think that's a good question. I think that you could categorize, you can choose what kind of network to use. Like, for example, there are direct interaction maps, but there are also like signal pathways, which could involve different types of interactions. In that case, you will need to involve like edge type, where you have to define heterogeneous edge types, where you need to differentiate a direct interaction versus maybe some like modulated interactions. But I think in general, making use of such kind of graph structures would give you additional information that the sequence level information cannot give. Yeah, that's a very good question.
If you have the structure information for two proteins, why is it difficult to know if they're going to interact? Like, can you run like a physics simulation?
Oh yeah, I think I'm about to talk about the docking part. But so yeah, you can run the simulation, but as I mentioned last time, like same story for folding, right? It's too expensive to do very precise modeling of dynamic systems with like.
It's just that they're like big. There's a lot of forces involved.
Yes, very big, and also there's solvent, right? There's like water molecule solvent involved, and a lot of the different forces, and it's even difficult to model like maybe 7 atom, like very small, like small molecules. Not mentioning these big proteins. So if you're interested, I think there's a recent work accepted to Science, I believe, that like people try to do kind of ab initio molecular dynamics for protein by kind of using machine learning to learn the force field for kind of dipeptide, like every pairs of amino acid. And so they kind of speed up the molecular dynamics, which at atom level into like dipeptide level interactions, and where they can manage the molecular dynamic process into like reasonable time frame. Yeah. But I think overall, yeah, I think it's always a problem of compute, because the time step, this time scale we're modeling with the simulation is just very short and transient compared to the process that's like long enough to solve the kind of the folding and binding problem. And the other thing is the accumulation of error. Like if we run it for too long, each step you have some error, and a lot of the simulation tools you accumulate the error. So at the end, it might just not make any sense if there's like imperfections in the force field or model system. Yeah. But I'm not saying that machine learning solves this problem at all, and we can see that machine learning is also only like, I think, alleviating certain issues or giving us the enough scalability and efficiency to make a good enough guess, but it's never like precise or accurate if we look into the errors and especially in the energy prediction task. Machine learning models are all very bad at the moment, I think. Cool. All right, so let's move to the task 2. So task 2, in this task we kind of know if two things already bind. We're tackling with the second level task, like where in the sequence, which residues in the sequence are important for binding. So it's kind of like we know that these proteins bind in a unique way. There might be multiple docking poles of two molecules depending on like the symmetries and the shape complementary in the surface. But in general, there are limited amount of possible docking poses. So this particular angle and direction that they're combined to each other. We call it docking pose, right? So for each docking pose, you can see that there are certain molecules that are in contact. So we usually can define a surface by taking all pairs of atoms within maybe certain threshold, like 10 Angstrom. So then we can basically highlight certain atoms or amino acid on the surface of the protein, and we usually call this kind of PBR, or protein binding residues, or protein binding site of each single monomer. So this task is basically like give me two protein, whether just sequence level or structure level information, can I predict their binding site in some degree, or predict where the interface is for their binding. Right. So the interface of the protein-protein complex are actually pretty small, so that's why the problem is reasonably challenging, because there are a lot of for interfaces with this small area, small sizes, you have so many possible interfaces that you can enumerate on top of the two proteins right there. If you try to look at all of them, it will be pretty computationally intense process. And I think that the methods try to kind of either using indirect information or like evolutionary information, or try to come up with smarter feature extractors that can allow like faster mapping or matching of the surfaces. And particularly, we can also call these interaction sites hotspots, and where hotspots can mean like even more stringent subset of residues, where we call them like the ones that contribute to more than 2 kmol binding. So you can see like usually only like less than 10% of the residues at the interface can contribute such a strong binding. So these hotspots are considered super important, and so it's very likely that if you alter them into something else, then you will lose binding or significantly lose the binding affinity. So one way of kind of guessing where the hotspots are is doing this mutational scanning, which we kind of mentioned a little bit in the previous, where you just kind of blindly mutate the residues at the interface, and you will observe like different amount of shift in binding affinity for this two system. So this kind of data can be used as a high-level like approximation of where the hotspot might be, and of course there are also like experimental data that can precisely categorize where the hotspots are by looking at their structures and make a guess of based on the distances and type of bonds that they're forming. But yeah, we can consider the task being trying to predict where these hotspots are, or where the interfaces are, given the sequence or some level of family information. So again, like earlier in the days, there aren't a lot of good structure predictors, so we kind of have to rely on sequence. And so there are different bioinformatics or machine learning models to try to like learn features, extract features and embeddings from sequence alone. And also like you can make use of the MSA information, which kind of like the DSM, but the DMS, but it's like naturally available data from evolution that kind of already tells us some information about which residue is important or not. So in the earlier days, this kind of information is used to make predictions of where the hotspots are. And particularly, for example, like one of method called Pythia, they kind of, again, like this is kind of what the data looks like, right? You give an input sequence and you try to predict this binary mask of whether this is a binding site or not. So in this example, like you can make use of either you can make use kind of the MSA transformer type of model, where you gather some homologies for the given query sequence, and we can aggregate their co-evolutionary information that will give us some both like a pairwise contacting information, and you could also use the embeddings to directly predict the probability of the site being a binding site or not. So the prediction stage, they kind of make use of two type of information. One is the sequence level neighborhood. So you can, at the sequence embedding level, you could, for each given target residue that you try to predict, you can look at the local neighbors in the sequence space, and also make use of the pairwise like contact map, such that you can also use attention to look into like global neighbors which are further down in the sequence but are close in the space. And by aggregating information at different levels, you'll be able to make these binary predictions, even relying on these single sequence and their evolution data. Of course, like if we have some structured information, that would be even better. There's ScanNet is kind of more recent architecture that people use to derive the binding site, and it's pretty good and pretty popular tool. What it does is, again, like making use of the geometric information, and you can precisely define different geometric graphs, which we talk about like a few letters ahead, and you can define it in both the atom level and amino acid level. And usually this atom level neighborhood is important in kind of deciding really precise and local interactions and information about the peptides. So you can see like a lot of the graph neural network models for protein-protein interactions try to, like zoom into an atom level in some degree. But also like the amino acid level information is also important. And so what this ScanNet does is it first using kind of a graph convolution neural networks in the atom level to aggregate the atom level representations, and then like apply another pooling to convert them into amino acid level information, and then do a second graph neural networks message passing in the amino acid level graphs. And the final outcome is the amino acid wise prediction of whether it's a binding site or not. And more recently, well, actually not that recent, but this is a very important work that came out where like geometric deep learning like first time show its strength in protein binding, in protein like interaction studies. Is what it's from kind of a group in Europe, where they come up with this. With some concept in geometric deep learning, they come up with this fingerprint of surface that can be used to derive some profile, again, like some fingerprints or features or profile of the geometries of the protein surfaces that we can use to inform where the binding sites are, for example, or just try to profile or categorize like different types of interface. So in the high level, I think Simon mentioned a little bit like when we represent proteins, we can represent it with kind of this stickgram where kind of we categorize the bounds and connected amino acid bound with the sticks and bars, and we care about the points, care about the kind of the amino acid either as a frame or as an atom, and we treat them as point cloud. And this is one way of representing the geometry. The other way is to kind of apply a mesh on top of these points. So for like maybe every 3 points or 4 points, you define kind of a small connected plane, and all of that plane, small planes come together to form a surface of the molecule. So this is kind of like the face. We're looking at the human faces, right? You have this kind of local features on the top of your face that shapes the 3D information about the face. It's kind of like the face for proteins, and it's kind of all 360-degree mesh applied on top of the protein. So why this is important is when we look at the structure-based or sequence-based information, right? If you look at the global similarity of sequence and structures, they can be very dissimilar. However, they can share very similar functions. And the reason is that even though they're very dissimilar globally, their local surface, especially the interaction surface, may have very similar patterns. So these local patterns can be categorized in one way, which is this meshed representation of connected area in the surface, and which we can call them like a fingerprint. Basically resembles a smaller structure information in the surface of the protein. So this is also an analogy is like in face recognition, like the earlier algorithm, they also try to kind of detect some key point and come up with meshes and derive some features that is already powerful enough to differentiate these faces with the other face and do matches between the faces. So the other important, not always true, but common belief about binding is if two things bind, their surface need to be somehow complementary, or kind of this shape complementary will help like smooth out the binding energy landscape. So usually if you have something with complementary shape, we consider this as like a good bias towards like very likely to be like binding or interacting. And therefore this kind of fingerprint can be used to also find symmetries or complementary of these local patterns. So we don't need to go into very detail, but in the high level, like in geometric deep learning, they have a lot of tools developed for meshed surface. And one of such architecture called MoNet, where you instead of defining things into this grid of like 3D grid, they define this local coordinate of surface. You can consider it as like some kind of change of basis or change of coordinates, where instead of like doing it in a Euclidean kind of Cartesian space, we have it like in the polar coordinates where you have the distance and angles or rotations. You have a different system of coordinate system to represent where the points where the meshes are. And they have this particularly defined soft pixels where we can turn each mesh into some coordinates or geometric information, and then we can apply like convolutions as we were doing in the 3D like Cartesian space. So yeah, just think of it as another kind of geometric GNN applied on a different coordinate system, particularly designed for surface. So with this kind of spatial convolution, we'll be able to get local features, which when we apply it on the protein, very similar architecture applied to the protein surface, we're kind of able to define these kind of local feature vectors or tensors that resembles a lot of the local shapes and curvature informations of the surface. And they found that using this small fingerprints of the protein surface, they were able to categorize and retrieve surface, compare surface with very high efficiency. So there are multiple different applications of massive. For example, you could, for a given pocket, a given surface, you could use it to classify what kind of ligand binds to the surface as a classification task. And you could also do a binding site prediction. If you have enough training data, you will be able to know what kind of service serve as the interaction binding site. And finally, they have this kind of retrieval type algorithm that enables very fast search between different proteins for potential candidates that can bind to a given service or given domain or given pocket. And this search of quantum complex is what makes it pretty powerful, and they show that it has a pretty good interaction, a pretty good precision in predicting things that actually bind and where they bind. So it's kind of like a way of, almost like a way of being able to like solve for binding or docking in some degree, right? Well, where for any given two protein structures, you kind of know where they align and how whether or not they align. All right, so the next step is to not just predict where potentially they bind, but precisely how they bind. And by saying how they bind, I mean the 3D conformation of the bounded structure of two monomers. Questions?
My question is, so does each protein have a unique, like only one binding site, or can it have multiple binding sites?
It can have multiple binding sites. Depends on the, again, the symmetry, and the shape complementary, and but not all of them has the same binding energy or binding stability. So some of them are just transiently interacting, but some of them are more stable. Yeah. So a lot of time in this kind of complex oligomers like capsid, which is the shell of a virus that involves like thousands or hundreds pieces of heterodimers or basically repeating of like 3 or 4 unit, you can see that there's very complex symmetry in the capsid, such that like some things can bind into very different patterns, but this pattern is repeated in symmetric fashion, such that at the end you piece together, you get kind of a ball-like structure. But when you zoom in, the interaction is like very interesting. So, so yeah, I think solving all of these kind of interaction problem and finding the most stable binding form or alternative binding form is very fascinating, and it's the area, the direction that we're going towards with task 3 and task 4. Right, so for task 3, now we're talking about docking, which is the process of, you can see, you can maybe if you make like a naive assumption that both of the monomer proteins are solid or are rigid bodies, meaning that in this docking process you only move them as a whole and you only change their translation and rotations such that they can piece together perfectly at the end. So this kind of rigid body with the rigid body assumption, which is solving the orientation and translation problem of like how you move one of them towards the other one. But in reality, proteins are not solid, so there will be also like local movement and dynamics happening when these two bind. So there actually are conformation changes when they form these complex, which make the problem even harder. So yeah, to precisely define the docking problem is to given the two separate proteins at its free form, we try to assemble the proteins in a way that kind of best resembles their stable binding form. And the task is to derive these 3D atomic model of the final coordinates to basically guess where the atoms are in this combined form or bounded form, given the initial free form of the two proteins. There's a slightly easier task, which you don't give the two proteins in the apple stage. Basically, you already know what the bounded stage are. So you know like when they bound, what the conformation these proteins are, because they move a little bit, right? But given this final stage, but only looking at the structure of each other, like you just predict how you rigidly move them so they can bind. Right? This is slightly easier task, but it's a challenging enough task that a lot of the paper is using this setting. And of course, there's a conventional method. As one of you asked, like there are a lot of different way of modeling, simulating this process with, for example, from the coarse grain level, you could use this homology modeling where you find very similar proteins and in the databases, right? If I have something very similar to me, then like the binding patterns from that protein might help me to guess the binding pattern of myself. So for the both party would like look into database and find a very similar subunit and find the subdomain and interface that are similar, and you can align them using these templates and try to make some initial guess, and then you can make some final adjustment. There's also like the ab initio docking, which like the molecular dynamics or quantum dynamics, where you kind of make some assumptions or computation of the biophysical forces, and you do this like really precise like simulation. And there are also like scoring functions where for a pair of proteins and their relative positions, you could give them a score based on like different types of potentials or energies. Some of them are exact, some of them are approximated, and just use it as a high level like approximation of like how strong the binding is. But like a lot of these are kind of working okay if you give it a very good initial guess, and they can be used maybe to also refine the docking, as what kind of AlphaFold did. So like one question is like if AlphaFold is able to predict folding, is it able to also predict docking? So with AlphaFold 2, this task is not as trivial as we thought. So one reason is, as I mentioned, the scalability of AlphaFold 2 is not really great because of these expensive triangle update and recycling, right? So it is actually, if you just apply AlphaFold blindly on like multiple chains, it may not be able to just learn that well from the existing crops of protein complexes in the protein database. So as a follow-up of AlphaFold 2, DeepMind come up with a kind of additional assembly algorithm that make use of AlphaFold predicted structures, but try also like augmented with additional like reinforcement learning algorithm to basically give you some way of assemble multiple predicted structure together into multimer. So this is what they call AlphaFold multimer model. where you have some initial guess with AlphaFold 2. And for each, you can consider like the final structure as this complicated graph, where like only the interacting or binding interfaces will have a connected edge, and each node in this graph is the subunit. So this kind of assembly process is basically predicting like which two nodes I'm trying to connect given the current assemble like substructures. So you can consider this problem as kind of like a reinforcement learning, because like each step you have some you use this predicted structure as guide, and you can train a policy to decide like which one that's likely that I will be add into my current partially assembled protein and where it aligns to. And each of the step, although you could enumerate all of them and choose the highest scoring one, but running this in the run like on the fly would be pretty expensive. So kind of using the similar reinforcement learning algorithm that's used in the earlier like what's the name? Like the AlphaGo in like the earlier version of AlphaGo, which is this multicolor tree search algorithm. We'll be able to kind of learn the optimal assembly path by seeing a lot of the assembled data and try to train with them. So we don't have to do this expensive like exponential like search during the assembly pass, and we kind of memorize the policy by training with this reinforcement learning algorithm. So before I talk about AlphaFold 3, I also want to mention a slightly different but equally important question of protein ligand binding, which is also like increasingly important besides protein-protein binding. So the only difference here is usually we, instead of considering two proteins, we're considering a small molecule that binds to like a small pocket on the surface of the protein. So like one side of the system becomes much smaller, and also like it's representing with a slightly different data architecture, which is usually like a 3D geometric graph instead of a amino acid graph or amino acid peptide. So the model architecture involves what we earlier talked about, like the SE3 equivariant geometric graph neural networks. And what in this equibind paper, they basically try to use the equivariant neural networks to directly predict the 3D coordinate. So given the initial like conformations and passing it into these binding architectures, it directly tried to predict the translation rotations of the small molecule with respect to the protein in one shot. And this is already like predicting okay or pretty well at the time. But so and recently, because there's a kind of this emergence of like these stochastic interpolate model like diffusion and flow matching, which we're going to talk about a lot from the next week on. There's improved version of equibind. Sorry, this is still equibind, but that's looking into more like details into the architecture. Basically, you have this two graph and this kind of indirect edges in between that you want to kind of learn a big graph neural networks on top of the receptor graph and the ligand graph. And with one shot, like transformation attention, you want to directly predict the final docking positions or basically the coordinates of where the small molecules are. So this task is, although it's kind of working okay in this version of equibind already, it is still quite a difficult task if you try to solve everything in one shot. So considering kind of the recycling thing in AlphaFold 2, right? And from AlphaFold 2 to AlphaFold 3, you kind of make use of this iterative refinement and multi-stage, multi-step, inference of the position. So moving from recycling to diffusion, naturally you kind of go from like a limited fixed step of recycling into kind of a more like theoretically grounded denoising process. So similar thing happens for the docking model, where kind of a more the later improved version called DiffDock from same group. They use kind of the diffusion model instead of the one-step prediction model to predict the translation rotations, just translations of the small molecule atoms. So the architecture is very similar, it's still equivariant architecture, but instead of doing everything in one shot, they try to model it in multiple steps of diffusion model. So this diffdoc proved to be like very robust, and it's giving like much better result in this kind of in this PDB bind data set, which is a commonly used data set for small molecule protein ligand docking. So yeah, so basically you challenge it with either a hollow stage crystal protein where you already know the bonded structure of the protein, and you just want to solve like where you put the small molecule into and the small molecule structures, or you challenge it with the apple stage protein where it's unbonded protein, and you just predict it with like AlphaFold or ESM fold, and this diffdoc approach, at least like from last year, it's like the state of the art of these docking problems. But again, like you can see that in this task, you were already given the structure of the protein in some degree. So, but even though you already have this information, you can see that there's still like a significant, RMSD like error, and so the top one accuracy with like smaller than 2, I think Armstrong error is only like 44%. Even with the structure information. So this task is like not easy at all. And so that's why what the AlphaFold debug makes it quite amazing that like AlphaFold is able to directly tackling the problem of both folding and docking at the same time, but and is able to outperform diffdoc on the protein small molecule binding problem, but also they show kind of very high success rate in a wide range of interactions, not just protein ligand, but also protein-protein interaction. Like if you see like the protein-protein interaction here compared to the AlphaFold multimers model, it improves with a significant fashion, especially on the antibody protein-protein interactions. It improves the AlphaFold multimer prediction by a lot, almost like double the success rate with this kind of new architecture that we talked about last lecture. And if you look at the protein ligand docking task, it's again like very impressive, like without the ground truth structure at all. And when you solve both of them together, it is able to outperform like diffdoc, which already you give it the structure information, and is kind of comparable to these like molecular dynamics docking models, where you give it a lot of information about the pocket. And so yeah, I think it's indeed a very groundbreaking step, and no wonder it kind of gives them kind of a Nobel Prize winning. But yeah, so yeah, I think we're getting much better now in the small, at least for the small molecule docking, and for the big protein docking, I think there's still some room for improvement. All right, so okay, I have like 7 minutes left. So so finally we. So finally, like the task of designing a drug is not just designing something that is complementary, or like designing just the shape of the protein, but also we want something that actually stays in a very stable form. So so I think task 4 is probably the hardest task, where we actually predict the energy landscape or the binding affinity for a given proteins, whether we have the structures or just the sequence. So even if we have structures, which are the methods that I'm talking about now, it is still a very difficult task. So formally defined, like a task here is for a given proteins, maybe with just the sequence or sequence and structure information, we challenge the model first of all, for the given sequence, for the given protein interaction surface, how well are we able to predict the binding energy or binding affinity directly from the given surface? However, like if you compare very different surface, right? Like for like A and B and C and D, this prediction may be trivial because these two input proteins are so different. So you can just blindly memorizing, like protein specific like binding affinity. So it may not really tells you about the real like biophysics in behind. So what the more challenging task here is kind of the affinity maturation task, where we look at the same target and same binding interfaces. We tackle the model with the problem, like if I mutate, doing some mutations or altering the one part of the binding, the binding party, by maybe making 1, 2, or 10 mutations, how much I change the binding energy. So everything has to be localized or normalized within like the same, with respect to the same target, and we just study kind of how well we can predict these local structures with some low throughput accurate data of delta G and kcat or KD, the association constant. So this kind of problem usually kind of have this data comes in pairs as like a wild type protein and a mutant. The mutant can be a single mutant or multiple mutant, and we challenge the model to like basically predict either the change of the predicted binding affinity, or you can solve an easier task of like just ranking the mutations based on your predicted affinity, or you want to precisely predict the delta G for making for each types of the mutant as well as the wild type. And there are limited, honestly, like very limited data for this task. So this is again like why it's hard, because like this precise delta G data, there are only like a small amount of databases. And one such model called GearBind from Mila, which just came out this year as well. They kind of introduced this pre-trainable graph neural networks called GearBind, where so they kind of introduced this geometric graph neural networks, which takes a pair of wild type and mutant, and you predict the structure of the wild type using fold-X, which is kind of a half semi-empirical or molecular dynamics, or I think semi-empirical force field model that predicts the structure of the wild type and also the mutant by doing some repacking of the sidechain. And they introduce this relational interface graph where, because you kind of know the docking position and structure already, you know where the interfaces are. So you take out the amino acid on the interface and you build multiple graphs. One of them is the bond graph, and also the other is the sequence graph, where you connect amino acid by their sequential information, and you also connect atoms and amino acid with spatial information, like the nearest neighbor graph. And they also do multiple levels of message passing, both atom level, like edge level, like bond level, and residue level. So it's a quite complicated geometric deep learning system. And it has two stages. The pre-training stage is done with this contrastive loss by kind of artificially generating similar like random mutations or rotamers. That's supposed to be like some of them we know that are very different from the wild type, and some of them should be very similar. And they use this contrastive loss to do a pre-training over the large databases of protein-protein complex, even though we don't have the precise, local mutational delta G data. And they fine-tune it with a small data set where we do have delta G for, and also with certain augmentation tasks. So after training, we could think of using these models. If it's able to predict this local change as well, we can consider using this model for a lot of high-stack applications. And one such applications is in silico affinity maturation, where instead of actually making a lot of mutations and testing all of them, you directly use the model to propose a few mutations that is likely to give you optimized binding affinity. And they're showing that the result is quite okay compared to like the fully like force field based model. And similarly, there's like UniBind, which try to solve multiple tasks at the same time. But they kind of have very similar intuition, where you build these interface graph neural networks, and both at residue level and atom level. And the difference is like you directly compute the tasks, directly compute like regression or classification loss, and you can do multitask learning here with different level of